Hello, everybody. Good morning and good afternoon. Welcome to today's webinar titled Mapping of the Brain, Spatial Profiling for Deeper Insights into Neurological Disease Mechanisms. My name is Sydney Panilla. I am with LabRoots, and I will be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar, Manisha Ray, PhD, Senior Director, Applications and Research Services, Vizgen, and Ioannis Mantas, MD, PhD, Postdoctoral Fellow, Karolinska Institute. Manisha, I will go ahead and hand it over to you first. You may now begin your presentation.
Great, thank you, Sydney. And thank you everyone for joining us today for our webinar on mapping and spatial profiling in the brain. I am from Vizgen. I am the Director of Applications there. And today I'm going to tell you a bit about Vizgen and the MERSCOPE technology in case you don't have background on that, and then highlight some new features we've added to give you more speed and flexibility at the same time over your experiments. So first, again, I'll talk a little bit about Vizgen, and then I will go into some data on spatial profiling in the embryonic brain. So in case you're not familiar, Vizgen is a Massachusetts-based company. It's a spatial biology company spun out of technology developed at Harvard. We're still here in Massachusetts, and our goal in spatial biology is to map as many tissues and disease states as we can to better understand how biology works and how we can intervene when it's not working the way it's supposed to. So Vizgen itself kind of grew out of the way that genomic technologies have been evolving over the past few years. About 15 to 20 years ago, with the advent of lower cost and high throughput sequencing, we developed the ability to sequence the genomes and transcriptomes of any organism at scale and have a much better understanding of the transcriptional and genomic states of many different organisms, tissue types, disease states, and so on. But we lacked sort of the individual understanding of what was happening at that level, which was really addressed by the advent of single cell technologies in the past 15 to 10 years. And those technologies let us see not only what's happening at a transcript or a genomic level, but at a per cell level, what each cell's identity is and what it's doing. So understanding kind of who the cells are in a tissue has been transformational in understanding what's happening in a tissue and in a disease state. However, all of those single cell technologies were done in solution. So the cells were removed from their native context in the tissue, which is not how any biology happens. So the advent of spatial omics is upon us, and Vizgen is one of the major players driving that. So with the ability to look at up to thousands of genes in situ, we have a much better understanding of not only what cells are there, what they're doing, but who else they're talking to and who they're interacting with. And those interactions underlie pretty much every biological process you can think of. So one area that in particular has taken advantage of the ability to look at these interactions is atlasing in the brain and central nervous system. Traditionally, neuronal tissues have been very difficult to study due to a combination of complexity, the current capabilities of technology, and the difficulty in accessing samples from human neuronal tissues. So the advent of spatial biology has really transformed the understanding of brain architecture. So we now know all of the cell types present in a normal mouse brain. Human brain atlasing is underway. There are atlases developed for other organisms. And once we have that kind of ground truth access, we can begin to understand what happens when diseases are introduced, what gene expression is changing, what interactions are changing, and how can we intervene to halt the progression of certain diseases like Alzheimer's disease. So MERSCOPE has really been at the forefront in enabling these atlases to be developed for several reasons. The MERSCOPE Ultra has a fairly large imageable area. We can look at entire mouse brain. We can actually look at up to 4 mouse brains in one experiment. We can also look at disease versus normal tissue in the same slide, in the same experiment. The MERFISH technology is extremely sensitive, which is important when you're looking for rare cell types. The technology was developed using mouse brain as the primary tissue, so it's very applicable to neuronal tissues. And all of this makes it a very translatable workflow. So the combination of features present in that were developed as part of the development of the MERSCOPE platform has enabled it to be uniquely suited to atlasing in neuroscience. The MERSCOPE is effectively the commercialization of the MERFISH technology, and it involves 3 major components. Sample prep, in which we get rid of the background of everything except the RNA in your sample. A very sensitive and error-robust design of probes to understand what's happening for up to 1,000 genes, and very high-resolution optics to enable us to get very highly sensitive, high-resolution data. So all of these things together, the large imageable area, the sample prep, the high error robustness, enable us to have extremely high quality data, and that's suited to developing these foundational atlases. MERFISH itself is basically a single molecule fish applied at scale. So the way this works is that we're doing in situ single molecule fish detection with a complex barcoding strategy that we refer to as multiplexed error-robust fluorescent in situ hybridization. We have a combinatorial barcoding approach where every gene gets a unique combination of probes, and each of those probes gives off a signal, and decoding those signals tells you the gene expression of up to 1,000 genes simultaneously in situ. So that's the science behind MERFISH. And in the past year, Vizgen has enabled even higher sensitivity MERFISH measurements through the development of the MERFISH 2.0 workflow. So in addition to the error robustness of the method, we've additionally incorporated RNA anchors to keep RNA in place. So if the RNA is degraded, you can still get high signal. And we've updated the barcoding strategy such that each signal generates more fluorescence than in MERFISH 1.0, which overall just increases sensitivity up to 10-fold. And we've seen that in many different tissues, including in a human brain. So the overall MERFISH or spatial biology mapping process always starts with panel design. And I will talk a little bit more about how that works on the MERSCOPE platform. The Vizgen is uniquely flexible when it comes to panel selection. We have a series of pre-designed panels. The neuroscience ones are highlighted here. And you can also customize a panel from scratch up to 1,000 targets. And we've recently added the ability to take a predefined panel and customize it in addition. So you can take an off-the-shelf panel and modify it to your research needs. Then you do your experimental design and sample prep, where you generate the MERFISH data. And then, of course, you go through data analysis, and then you can get to your biological conclusion. So this process has been used to great effect. MERFISH and MERSCOPE are the most published methods for doing single cell biology at the spatial level. We have over 300 publications that use MERFISH and MERSCOPE. Those publications cover over 50 different tissue types and represent work from hundreds of different researchers all over the world. In particular, we have had great success in working on brain atlases, 4 of which are highlighted here. There have been about 13 of these published using the MERSCOPE technology, and more are coming. In addition to whole brain mapping, now we're looking at mapping other parts of the central nervous system. So this past month, a paper was on the cover of Cell that used MERSCOPE to map the mouse olfactory receptor system. They basically did MERFISH for all of the mouse olfactory receptor genes in both the olfactory epithelium and the olfactory bulb, the structure right next to it. And they were able to show that there's a really clear spatial pattern of the receptors in both regions that are basically mirror images of each other. And they were able to couple this with functional measurements to show certain odors lit up certain parts of the epithelium and the olfactory bulb. So really cool paper that really gives a detailed foundational model of how the olfactory system works and how it functions in response to actual sense. So I'm going to spend a little bit of time talking about our updated panel design strategy that gives you the ability to start working more quickly and adapt those panels as your research question adapts. So Vizgen's panel design strategy, we have a series of pre-designed panels that allow you to basically have a starting point for your work. Making a panel can be a very time-consuming process. It can be difficult to make 1,000 decisions for every biological question you have. So our pre-designed panels offer a really nice way to make 815 of those choices up front and not have to spend a lot of time on panel design. Now, everybody has their particular gene of interest, and if that gene isn't in the panel is not useful for your question. So we've recently added the ability to customize those pre-designed panels. So you can add up to 100 genes of your choosing into the pre-designed panel, and which enables you to customize it to your specific research question. This can be really helpful if you're working on, say, a xenograft model, or if you have multiple disease indications, say you're working on both Parkinson's and Alzheimer's, you could have different panels added on to the base human brain panel to have multiple groups basically using the same panel. And this gives a really nice way to standardize across lab. We can be sure everybody's using the same genes, gene list, the same method for a bunch of different research questions. And that enables really nice reproducibility on the type of atlasing scale experiments that are so commonly done with our technology. The full list of panels that we have is shown here. In April of this year, we basically doubled the number of panels we had available. And by the end of this month, we will have quadrupled the number of panels we have available. So you can see from this list, we have panels that cover a vast range of tissues, disease states, and functional states in both human and mouse. And today I'm specifically going to highlight some of the work we've done with the mouse transcription factor panel in both the panel capacity and the add-on capacity. And what this means is, and here is the mouse transcription factor panel is highlighted here. The entire mouse transcription factor ohm is about 1,500 genes that we split into 2 panels, which can then be further customized with add-ons. So our 2 panels are called the transcription factor panel A and B. One is more focused on embryonic stages of development, and the other is focused more on mature mouse tissues, so that between the both panels, though, you have all transcription factors in the mouse covered. And this enables you to look at many different functional states within the mature or developing mouse, which means you can see how different tissues are developing across time and across space. Similar to the olfactory receptor paper, we could see across space how different olfactory receptor genes are laid out. And that can then be used to understand which developmental programs, which transcriptional programs are turned on and off in the normal state and in the disease state. And much of this work is very applicable to humans, where you can't necessarily study every transcription factor in every tissue. So there's a really nice translatability of this panel between human and mouse. The actual distribution of each panel is shown here. You can see we cover a wide range of transcription factors that are known to be expressed in certain cell types, as well as transcription factors involved in different processes. So sort of the cell type and the signaling pathway associated. And all of our panels are tested on the relevant tissue type. Here we are seeing panel A and B tested on mature mouse brain, and we see the expected expression of different regions of the brain with both of these panels. But again, if a panel doesn't have your particular gene of interest, it's not very useful to your research question. So now with the ability to add up to 100 genes into the panel, you can customize these panels to be specific to your research question. And here we'll show an example of how we've used the customizability of the mouse transcription factor panels to look at both cell typing and transcriptional regulation in the same tissue. Before we did anything else in developing these panels, we wanted to make sure that we could maintain our extremely high quality. So here we're looking at technical replicates of the panels with 40 added on genes and 100 added on genes. And you can see we get very high correlation between replicates with the additional genes and even between different numbers of additional genes. And you'll see on the right side that we don't see any change in transcript counts. So we are confident that adding on these additional genes doesn't change anything about the biology and maintains the high sensitivity of the MERSCOPE platform. So you get the added flexibility without any hit to data quality. To demonstrate the capabilities of these, we developed the transcription factor panels and added a spike in of 100 cell typing genes into both panel A and panel B. Again, together, these represent all the transcription factors present in the mouse. We then used these panels with the corresponding add-ons to look at both mature mouse brain and developing mouse embryo. And then we analyzed it to see what genes were co-expressed at different stages and different regions of the mouse brain and the mouse embryo. As expected, the data quality is quite high. We see really excellent correlation between technical replicates. And when we specifically look at the common genes, so the spiked-in genes on both panels, we see, again, excellent correlation. And those are the genes in red, are the common genes between the two panels. And as expected, we see the expected cell types present in mature adult brain, which we verified using single-cell RNA-seq to define those cell types. One of the really cool things that having these add-ons enables us to do is expand our analysis capabilities beyond exactly what's in the panel. So because we have the same add-on panel in both transcription factor data sets, we can use those as essentially an anchor to combine the data sets and infer gene expression for genes that weren't necessarily present in the panel. So on the left side, you can see that the detected expression and the inferred expression are nearly identical for all the genes that we looked at. And now with this larger data set, we can do bioinformatic analysis to identify genes that are co-expressed in different tissues spatially across the brain. And the results of some of those are shown on the right. So this is an unbiased look at what genes are lighting up the different regions within the mouse brain. And this, of course, shows canonical markers, but it also shows genes that are expressed alongside those canonical markers and can be a nice tool to understand what could be additional functional targets or additional targets for drug targeting and so on. We repeated this process in 3 stages of mouse embryo. P14 is shown here. And again, we see very high data quality, and we see labeling of all the major cell types that you would expect to see in the developing mouse embryo. We've pulled them apart a bit on the bottom of the slide here, and you can see we're getting really nice resolution even at the sub cell type level. So we can see the difference between granular keratinocytes and basal keratinocytes, for example. And because, again, we have the same spike in both of these panels, we can apply the same approach to use those as anchors and combine the data sets to infer gene expression and then identify gene modules. Oh, I'm sorry, I realized these slides were not advancing. There's the embryonic data, and there is the inferred data. And here's the result of some of these gene modules. One of the cool things we can see is really single cell level expression of different genes. So for example, our unbiased analyses pulled out genes that were associated with different layers of skin. And you can really see at the single cell level that one module is labeling an outer skin layer while the other is labeling an inner skin layer. And we also can see this nice gradient of gene expression of genes in the bone marrow. Here's even more zoomed in of the developing embryo. We also see really nice architecture of the brain in the developing mouse brain, and you can follow this through the different stages of mouse development. So to summarize, our add-on panels allow you to take our many pre-designed panels and then customize them for your specific research question. Here we show by adding cell typing markers into our transcription factor panel, we can look at the entire repertoire of transcription factors in both developing and mature mice and identify new genes associated with different developmental programs. So to wrap up, the ability to have this many pre-designed panels with add-ons gives you unmatched control over your own research. We can take the effort away of having to design 1,000 genes from scratch while maintaining the ability to tailor these to your research needs. And we've specifically focused on mouse here, but MERFISH can be applied to any tissue type. And again, we have publications on dozens of different species, tissue types, etc. So I'll end just with a preview of things that we're working on in the future. As we noted in the beginning, nearly every biology question is a spatial question, but spatial isn't only in 2 dimensions, it's in 3 dimensions. So we are working on adapting the current MERSCOPE Ultra platform to be able to analyze tissues in X, Y, and Z and understand in 3 dimensions how cells are interacting and what neighborhoods look like, who they're interacting with, and so on. And we will be showing a preview of that at the Human Cell Atlasing meeting that's coming up this weekend. So with that, hopefully I have given you some good background about the Vizgen MERSCOPE capabilities. And with that, I will hand it over to Ioannis to talk to us about some cool atlasing work he's done with the MERSCOPE.
Hello, everyone. Thank you for joining. Actually, can I make it live here? Like, could you see that? Okay, so perfect. Thank you for joining. So as Sydney said, I'm a postdoc right now in Karolinska Institute. And I'm going to present you, like, as the title says, like spatial profiling of dopamine neurons and related to Parkinson's disease. So just to give you a brief overview of what I'm going to show you, first of all, I'll try to convince you why MERFISH is important to do that, like in research of Parkinson's disease, why it's important to identify dopamine neuron subtypes. And then I will show you like how we did it to find vulnerable cell types. And at the same time, I'll try to show you how this technology actually helped us, allowed us like to find these cell types and make mouse lines that we could target the cell types and study the behavior. So it has like also like an expansion like to the behavior that is also always a question in neuroscience research. So for those that are working with Parkinson's disease, obviously they might know substantia nigra. That is the main area that is studied in Parkinson's disease. And I will go like through the history of substantia nigra briefly to show you like how we advanced like from just pointing it out like as a black substance in the brain. Not that now we know that it has like numerous cell types and different cell types that project to different brain areas. So the guy, the physician that first described substantia nigra was Felix Vic Desir. Just also a fact about him. He was like the doctor of Marie Antoinette in France. And in his work that I show you here, he had like very nice illustrations of the brain, the human brain. And you can see very nicely actually that he spotted like this black substance here. You see like that there's a brain area here that is mostly like has dark color. And the reason it has dark color is because it has a natural pigment that is neuromelanin. So later, there were many researchers and prominent researchers also at Karolinska that they dissected out the dopamine neural cell types. So now we know that this black substance has dopamine neural cells that we know that they die in Parkinson's disease. And also one thing that we know is that they die in a progressive manner. That's kind of important because it seems actually that there are some cells that are more vulnerable than others. And always there was this question. So do they belong to one cell type that just dies randomly? Or there are different cell types that display different vulnerabilities across the disease? So what we know is that there are many dopamine neuron areas. That Felix Victazir mentioned, the substantia nigra, is that one that is located here as green, as you can see. And there is also a medial area that's called VTA that we found out later in history, that we know that this area actually is resistant to Parkinson's disease. The vulnerable one is the substantia nigra. So already we have an implication that there is like some kind of vulnerability between dopamine neural cell types. And what we know as well is that the dopamine neurons that die in substantia nigra, they contribute to the appearance of these symptoms. So bradykinesia, rigidity, postural instability, and resting tremor. So these four are the cardinal symptoms / signs that define Parkinsonism. But nowadays, we know that Parkinson's disease affects dopamine neuron substantia nigra, but substantia nigra is not a uniform population. So there are like these are the most important studies, as you can see, the Salmani et al. At 2024 and the Gertner et al. At 2024. Both of them were published in eLife, that they did like single cell RNA sequencing to disentangle like the different cell types in substantia nigra and VTA as well. And as you can see here, there are many cell types. And we know that those are located in substantia nigra, those that are colored with blue here and here, that they have these transcription factors called SOX6, as you can see here. But it's not only in the mouse, also in humans, there are like several single cell studies. Two are the most prominent, so the Siletti et al. And Kamat. Kamat gained a lot of attention recently. And here they also pinpoint the same, that they were done also in Parkinson's disease tissue, and they found out again that substantia nigra is that one that is the most vulnerable. However, the problem here that exists in human tissue is that if you take tissue from Parkinson's disease patients, you always take like from later states, because like it's like from postmortem tissue, so the patients already have advanced disease. So we don't know with cell types if there is like differential vulnerability across the stages of the disease. So for us, was this main problem, since we know, okay, Parkinson's disease is composed by these four cardinal symptoms that they appear at the diagnosis of the disease, but we know that Parkinson's disease, it has a lot of symptoms and signs that occur throughout the disease, and some of them appear very early on, so even decades before the diagnosis comes. And so then having like the single cell data and this variability of the signs and symptoms, there is this theory. And this theory that maybe different cell types that they are located in substantia nigra contribute to different types of signs and symptoms. So for instance, here there might be like cell types that contribute differentially to rigidity or postural instability or tremor or even bradykinesia. So our question in the lab was that, okay, prodromal Parkinson's disease is that one that is defined before the diagnosis. And in order to make diagnosis of the disease, you need to have this criteria, that you have bradykinesia and at least one other motor sign from those that I mentioned, those four that I mentioned. So it seems actually that you might have bradykinesia early on in the prodromal states, but you're not fulfilling the criteria of Parkinson's disease. So you might have some kind of loss. So you might have lost some cell type that it's very vulnerable in the early stages. So for us, was like the aim of the study is like to find like the dopamine neuron subtypes that are vulnerable at the early stage of the disease, at the prodromal states, and define also, like, since we will pinpoint them and that there's this type of cells, define their role in the behavioral role, how they contribute to the symptoms of the disease. In order to do that, though, that's like a main problem of the Parkinson's disease research, is like that most of the time the models that we use are not really progressive. So they trying to mimic the disease, but always they are trying to have like a snapshot of the disease, as I can say. There is, though, a model that recently gained a lot of attention, even though it was described first in 2007, as I can say so here, is this MitoPark model. This is based on the deletion of a transcription factor that is nuclear encoded but goes to mitochondria, so transcribes mitochondrial DNA. So creating a conditional knockout in dopamine neurons of this transcription factor creates like dopamine neurons that they don't have healthy mitochondria, and they degenerate. And interestingly, this model displays progressive dopamine neural cell loss. And as you can see here, and that's like stainings of dopamine axons and dopamine neurons in substantia nigra, that up to 43 weeks of age, they almost lose everything. But the disease starts like we see that there is the early loss. Starts like from around 10 to 12 weeks of age. So what we did is we decided to use this model and use different stages of this model, like some that there might be prodromal, some that might be that prodromal is like around 8 weeks, so before the symptom starts, some that they are at the early symptomatic stage, that is like 10 weeks, and some that is a bit later stage, but still middle stage Parkinson's disease, that's 16 weeks. And since there are like plenty of single cell studies, as I showed before, we designed a 140 gene marker MERFISH panel that we found like the genes that they are mostly like separating all these clusters that we want to see. And then we applied to this tissue. And the thing is also that since we are interested in the spatial information, so we try to capture the substantia nigra throughout the anterior posterior levels. So then we try to capture like 3 different levels. So, and as you can see here from the numbers of the animals and the levels, that's a substantial amount of tissue. And in order, so I will show also some technical details since we are talking about MERFISH. So what we try to do in order to increase the yield, let's say, of our information, so we use this approach. So as you can see here, that's like a Vizgen slide. That's the small one. It has 1 by 1 the area of where you put the tissue. And as you can see here, what we did is like we put the 3 levels, but in this distribution. So we cropped out the tissue that we don't want, so we don't care. So since we focus in substantia nigra, we don't really care about the surrounding brain areas like the hippocampus or upper midbrain and stuff like this. So by doing this, we managed to have like a huge data set. And this huge data set actually is here. You can see like an example tissue section, how it looked like the cropped one. And we managed to have like from the whole data set almost 1 million cells, and that they have this clustering distribution. And from all the cell types, we found actually that the one that is interesting for us is this pink one, as you can see here, that expressed like very nicely the dopamine neuron markers. So this seems to be the dopamine neuron cluster. And then what we did later, we took this cluster, we subclustered it, and we nicely dissected out the different dopamine neuron clusters, and almost resembled the 2 single cell studies that I mentioned before in the mouse. So it was very nicely, the resolution of the genes, even though it's a low number, gave us like a very nice resolution to see the cell types. And not only that, we validate that these are the cell types from the area that they are located. So that was also very nice, the spatial information. So we could like define like ventral substantia nigra clusters, dorsal substantia nigra clusters together with VTA clusters, and also those that they are located in the very lateral part of substantia nigra. That is also gaining attention as a population, this one. And also like they're also located in VTA as well. And having everything all together, so now we could see actually which of these clusters is the most vulnerable since we have the MitoPark tissue. And to do that, we used a statistical analysis called MASK, so that this one actually calculates the probability of having a cell cluster in its disease, like grossly what it represents. And here, actually, those that they are minus basically is the most vulnerable. Those that they are like plus is the most resilient population. And we did it in all the different states separately, and also in overall mitopark, which is most depleted. And it's here I positioned the clusters according to vulnerability, so the lowest to the highest. And here you see that this DAN2 cluster is that one is the most vulnerable. And in every stage of the degeneration was that one that popped out as the most vulnerable. And even taking the overall mitopark seems to be again as the most vulnerable cell type. And we had also some clusters that they are like the DAN13, for instance, and DAN11 that consistently were in the resilient part. So we managed, we managed also like to characterize how their fraction over the total dopamine neurons, because that's the actual vulnerability marker, goes over the disease. And you see very nicely that this DAN2 cluster declines steeply as we go over the disease progression. A DAN3 cluster was also pretty vulnerable. So for this reason, we tried to compare these two together. And it seems, and then we did like some kind of half-life analysis to see like the decay, because it's like studying the decay of degeneration. So, and it seems actually that the DAN2 cluster was like had lower half-life than. So then you lose 50% of the cells earlier than the DAN3 cluster. And then we also found how they are distributed in the tissue, and you can see very nicely all over the stages of the disease how nicely the orange cluster, this DAN2 cluster, is depleting along the way. And then the question is like, okay, this DAN2 cluster, what is it doing? What are the markers of this DAN2 cluster? So then we could define actually very nicely a novel, I should say, markers of the resilience and the vulnerability. And as you can see here, there are like some markers that they're very nicely located in this cluster that are resilient or vulnerable. And we have the SEMA5B, POSTEN, that is the resilient, and ANXA1, LDH1A7, that they are located towards the vulnerable territory. But the marker that defines the best, the two markers that define the best, again, the DAN2 cluster is those two, ANXA1 and LDH1A7. So LDH1A7 is a gene that is not really, actually is unique to mouse, so it doesn't exist in humans. So for this reason, we decided to study the ANXA1. Annexin A1 is the name of the protein. And we try to validate that. So because there is like very nice probes and antibodies to study that in tissue. And we could see actually, again, in the midbrain, how it's located. And it's like almost identical to what we saw in the MERFISH. So it's located ventrally in the midbrain. And we could map it like throughout the anteroposterior axis. And also like if we plotted like the ventral dorsal position of the cells, they are located mostly in the ventral substantia nigra. And then we validated that the same results in mitopark mice to see that that's true. It's not just some kind of artifact. And it seems actually that with RNA-scope, we could see that this ANXA1 is the yellow one here, the yellow gene, is depleted in the early stage of mitopark. And this purple one that is the conventional, I would say, vulnerability marker, LDH1A1, seems to be it is vulnerable, but ANXA1 is a subtype of the LDH1A1 that is more vulnerable than the gross LDH1A1 population. And then we compared that with vulnerability of the total substantia nigra, that is like the major vulnerable cell type, let's say, as we knew from conventional Parkinson disease research. And it seems actually ANXA1 can be best vulnerability marker, the best vulnerability marker. And then, okay, since we have ANXA1, we try to test it. Okay, in mice looks nice, but is it happening in human as well? So for this reason, we took like Parkinson disease patient tissue, midbrain, and we tried to see if ANXA1 is there. And it seems that it's there, even though it's not very abundant. So as you can see here, the blue one, this is RNA-scope. The brown thing is the neuromelanin, is this pigment that I mentioned before. Pink is the Th, is the general dopamine neuron marker. The blue one is the ANXA1. And you see that we have populations that they are like pretty abundant in ANXA1 mRNA, and some that they had like few transcripts. Nevertheless, we found that the ANXA1 population, the ANXA double positive neurons, the Th and ANXA1, it seemed to be preferentially depleted in Parkinson's disease tissue, as you can see here from the statistics as well. And interestingly, because as I mentioned that the tissue that we get is mostly late stage Parkinson's disease, we try to see in the control brains as well, if we correlate with the age, the AXA1 population, it seems that it's declining. So that because usually control brains, they have like subclinical Parkinson's disease, because also it's a disease of aging, of normal aging even. So it seems that actually AXA1 can predict even mild disease. So, and then back to mice, what we did is like we designed an AXA1 mouse line, AXA1 flip mouse line, since we found this population, and we tried to label it genetically. And we did it very nicely, actually. The mouse worked extremely nice. We were super lucky that this AXA1 was restricted to dopamine neurons. And we also managed to map the projections to the main output region of the dopamine neurons, that is the striatum, that's also the region of the most of the Parkinsonian symptoms that we see. And what we saw is actually that AXA1 projections in the striatum, they are mostly located in the very dorsal part of the striatum, the very dorsal tip of the striatum, just below the corpus callosum of the mice. And also slightly enriched to the dorsomedial part. And then we want to see the behavior. So if you block AXA1 mice, do they recapitulate the Parkinson's disease features, or is it just the general dopamine neuron depletion model? So what we did, then we took this AXA1 flip mice and we expressed a tetanus toxin, a flip-dependent tetanus toxin. Tetanus toxin, just a very brief introduction to that, what is this? So it's a toxin that basically ablates neurotransmission. So as long as you express it to these neurons, they stop releasing neurotransmitter. So basically, these mice don't release dopamine from this cell type. And then we compared actually these mice with this mitopark, different mitopark mice. And we compared it in what behavior, so just open field. So we didn't really do some very sophisticated thing, but in the open field, and that's it, because it's the main output that most researchers are validating that the Parkinsonian phenotype. The only difference here is that we recorded from below. We used the deep lab cat in order to track all the paws of the mouse and the snout. And I will show you why we did that later. So, but first of all, we just measured like gross mobility. And if you see just the AXA tetanus mice, you see actually that they decrease their speed and angular speed, and also slightly increase mobility. But it's not like something that is like an overt Parkinsonian phenotype. And if we compare with the other two Parkinsonian mice, they resemble the mitopark early, and not the middle one. I repeated that. So the middle one is not late stage Parkinson's disease, but you see that already the middle has like a feature that is increasing immobility a lot. So that the mice spend more time like immobile. So this phenotype actually resembles a bradykinetic phenotype, pure bradykinetic, without the rest of the three motor signs or symptoms that I mentioned before. And in order to go one step further, and that's then now I should say that's why we did record from below, is that there are like several techniques nowadays that you can you can do behavioral segmentation, and you can have like a sophisticated analysis of behavioral mouse behavior, mouse behavior. I'm sorry. And here we used VAME. This is an approach that we use VAME because the VAME is designed like with cameras from below, and we managed to dissect out the different behaviors. So what we had is like different types of walking, different types of rearing. So behaviors that we couldn't really tell the difference, but the AI-based software could tell. And it's like doing sequencing, but for behavior, I would say. And then what we did is like we took all these behaviors and ran a PCA. And I would like to tell you now to ignore the purple, the red, and the pink here. So just focus on the AXA1 tetanus, that is the yellow, and the blues, that is the mitopark. And what we see here is that the controls are located mostly in the right side, so the right side of the PC1. But you see that the AXA tetanus, they're going to the left, so they're affected mostly in the PC1. But the PC2 score is describing more the late states. So it seems actually that AXA1 tetanus is positioned somewhere in the middle between the middle stage Parkinson's disease and the early stage Parkinson's disease. And if we look the specific behavioral motifs that they were affected, and we arrange them according to speed, So those that are in the very top part of the graph here are the high-speed motifs. Those that are low is like the low-speed motifs. You see that AXA1 tetanus affected, the AXA1 tetanus mice had like motifs affected that they're on the high-speed range, while the low-speed motifs are not affected at all. Like if we see the rest of the Parkinson model, or the rest of the data with the Parkinson disease models, you see that the mitopark middle has severely affected the mobility motifs. So those are their low speed. So it seems actually the AXA1 tetanus makes like a phenotype that looks like bradykinesia prior, rather than having rigidity, akinesia, or the rest of the symptoms. So what we did here, and that's the important the conclusion, is like we defined and NeurLPD vulnerable dopamine cell type that contributes to parts of the bradykinesia complex. And how we would frame it in the patient spectrum of the disease progression, it's like that we think that AXA1 dopamine loss is located somewhere here. So it's just before the diagnosis of the Parkinson disease. So it's like a motor syndrome that is like subtle. The patients do not really see it themselves until they go to the doctor. So the future research should, we are trying now actually to resolve this part. So how AXA1 dopamine loss might affect a very early prodromal states, that is like even before the bradykinesia, or if it contributes to any non-motor symptoms. But that's the main overview of what we did. So, and here now we'll finish my presentation, and I want to thank all the collaborators and the co-authors on this, because that's a preprint, what I showed you here. And mostly Alessandro, Vasiliki, and Ines, that we work closely together. So these are the main people that worked intensely in this project. And of course, ASAP, that is funded us, and also SSMF, that's also funding me. Yeah. So thank you.
All right, fantastic. Thank you both for the very informative presentations. We will now go ahead and move into the Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. All right, let's go ahead and jump in here. The first question that we have is for Ioannis. It says, why is VPA not affected in Parkinson's?
Okay, that's a very, very hard question to answer. I mean, and then it's like, so VTA, I guess you mentioned here because you're right, VPA, it's ventral tegmental area. So the same question can be applied the other way around. So why substantia nigra is more vulnerable in Parkinson's disease? There are many theories still. We have no clue why is this happening. The major theory that was back in the days introduced is like that there is like a marker that VTA expresses mostly, that is cal binding, that is mostly like, it's a protein that is handling the calcium in the cell, because calcium is considered to be one driver of the cell death in Parkinson's disease. But I don't think that that's the main reason, because you see that the vulnerability goes beyond the expression of that gene. So cal binding is traditionally described to be the one that defines resilience, but we think that it's something more than that. Another theory is like the distribution of the axons. So VTA, that they have more narrower, I would say, projection fields in the striatum. So then they are not so energy demanding. While the substantia nigra ones, they have very broad axonal fields, and these are energy demanding, and because of that they die. So still we don't know. And then and one other question that I get many times whenever I present that these data is that why AXA1 is something that is related to vulnerability. Still, we cannot answer. What I can say is that AXA1 is something related to inflammation. It's a gene that has many functions, but one of the functions like as a response to inflammation. So it seems actually that these neurons located in substantia nigra, I think they fire in a different manner than those in VTA. So they have a bit like, as we call it, pacemaking firing rate. So that in the same pacemaking, it seems that they have like they fire like in a phasic response, let's say. So they constantly fire, while VTA they can be pausing, let's say. So that means that they are like heart cells, let's say. So they are trying to fire all the time. So they have like a frequency of firing that's all the time there. So that makes them like to be more vulnerable to oxygenation, let's say. So then they are more prone to accumulate radioactive oxygen species, or stuff like this. Still, though, it doesn't really explain the vulnerability in general, because, yeah, I mean, so it's something that is ongoing on research. So I cannot really give a clear answer. So I gave you like few topics of what we think that's happening, but yeah.
Okay, fantastic. The next question that we have here says, does MERFISH 2.0 use different probes for different RNAs simultaneously?
Yes. So it's actually dozens of probes per RNA. That's where the highly multiplexed and error robust barcoding comes in. So we put 20 to 50 probes per gene on each gene that you're interested in, and we can look at up to 1,000 probes or 1,000 genes at the same time. So all of the genes are looked at simultaneously. Each of the genes has a different combination of probes, and the barcoding of that probe, it translates to what gene it is. And when you do a MERFISH experiment, you get what we call a code book to decode the barcode to the gene itself.
Okay, great. This next one is for Ioannis. It says, how did you decide on 140 genes? What considerations do you have for going bigger or smaller with your panel?
No, it's not like something very. So we went to the least possible, actually, because I mean, to have a custom panel also, like, it goes a bit more expensive, let's put it that way. So, but we thought that still, like, because since there are existing single cell studies, and we knew that 140 genes, it's fine to dissect out the populations that we're interested in. So we didn't need to go further. So it's not like something we could go further. But also the problem is, like, sometimes if you go further with single cell studies, there might be some probes that do not work. So we checked one by one all the genes that we like put on the list that they have nice expression in Allen Brain Atlas, for instance, so that they are not like, because there are a few genes that you see as markers single cell, but when you see the in situ looks like so and so. So that can be many reasons. So so that was the main reason. So if you have fewer that that expressed well, I think it's better than just go higher, and yeah. So that was the reason.
Okay, great. Manisha, this next one is for you. It says, how do researchers decide between using a custom panel and a pre-designed MERSCOPE panel for neuroscience studies?
It really depends on your question, I would say. I think the easiest course of action is to always look at our pre-designed panels first, because then if the genes that you're interested in are in that panel, and most likely they are, then you can go with that and not need to have any other barriers between starting your experiments. So those panels are off the shelf. So as soon as you decide that you're ready to order a panel, it can be shipped out to you. Whereas the custom panels are custom made, so the shipping time on those is a little bit longer. So I would say if the pre-designed panel fits your research question, it's easier to start there. But even if it is not a perfect match for your research question, one of the cool things about our panel design portal is that you can use any of our pre-designed panels as a starting point for designing your own panel. So you can take out the genes you're not interested in and add in your own, and not have to make all 500 or 1,000 decisions, but just pick the ones you're interested in and add it to a pre-existing gene list.
All right, Ioannis, this next question is for you. It says, how consistent was the vulnerability signature across the different Parkinson's disease models examined?
When you say, that's an important question, actually, but it depends how you say Parkinson's disease exam. If you talk about the mitopark themselves, those that we use, that was extremely consistent. Actually, I couldn't really believe that was so consistent. And the nice thing about this, because okay, when you do in situ and you count the cells, you might use like a semi-automated like software to count the cells. In our case, we used the Mars, for instance. The nice thing with the Vizgen software was that it was fully automated, because you use this cytoplasmic marker and the DAPI that is also for the nuclear. And then I didn't count the cell myself. So then it was like the data was like just fully automated. When I saw the vulnerability, then I believed it. I said, okay, that's how it is. Cannot be like something else. But if you're talking about different Parkinson's disease models, that's also a very important question, because what we use here was a very specific mouse model that is relying on the mitochondrial part of the Parkinson's disease. Because Parkinson's disease, one of the mutations that is known is like this Parkin and pink that affects mostly mitochondria, but the most common one is alpha-synuclein. And mutations that they are associated with alpha-synuclein pathology. So in our preprint, we use MitoPark. That was nice, like to pinpoint to track the degeneration. But we also used like 6-hydroxydopamine, that is a toxin model. But also another model that is like autophagy based, that has also some kind of Lewy body-like pathology. That's called like ADG7 knockout. Not global knockout. This was like dopamine neuron conditional knockout. And also there we found that ANXA1 are like preferentially lost. So it seems to be just general. I don't know what's happening with alpha-synuclein. It's tricky when you work with alpha-synuclein itself, like with viruses or transgenic models. So we didn't really use that. But as long as like when it comes to toxin-based models or like autophagy-based models, ANXA1 was again preferentially vulnerable.
Okay, Manisha, this next question is for you. This one says, does the tissue size matter to be able to look it on in MERFISH?
The tissue size doesn't matter. It's however you do your histology is sort of how you define your tissue size. You can put multiple sections during histology onto a slide. But the maximum imageable area on the system is 3 centimeters squared. However you use that 3 centimeters squared is very flexible. And depending on how adept your histologist is, you can fit more. You can really pack in that 3 centimeters, or you may be able to fit your whole tissue on there.
Okay, well, we are reaching the top of the hour here, so we will go ahead and wrap up with this one final question for Ioannis. This one says, could ANXA1 expression potentially serve as a biomarker for identifying vulnerable neurons before overt neurodegeneration occurs?
That was like the main, that's a nice question. That's what we also try to see right now, actually. I would say it's not so easy, because it depends where you measure it. You measure ANXA1, because you could measure it either in the blood or the CSF. So in the blood, so ANXA1, there are many, actually macrophages, I think they have a lot of ANXA1, so maybe it's a bit hard to extrapolate something that's happening in the brain through the blood. But when it comes to CSF, okay, ANXA1 is like pretty highly restricted to dopamine neurons, but at the same time, it's also expressed in several tissues. It's not in neurons so much, it's mostly like in pia and the pendymal cells. And that makes a problem into that. So then, because you might find it in the CSF that is not really coming from dopamine neurons themselves. However, so our idea was that not to use ANXA1 itself, but rather use like a similar approach as we use in the mice, like to do behavioral segmentation to find subtle motor changes, to do something like in humans, or use an approach like a very advanced like MRI that you could somehow pinpoint like the ventral substantia nigra where ANXA1 are located, and say, okay, you have lost cells there, so then you have an early marker of like Parkinson's disease. I don't know if ANXA1